Everybody. Angel Phan is going to be presenting. Her advisor is Trish Bainham, and she'll address everyone after Angel's presentation.
Hello everyone, my name is Angel Phan, and I am a senior biology major undergraduate student at St. Edward's University. Today I will be presenting my work in determining the distribution of class 1 integrons in multiple drug-resistant Salmonella enterica, cerevar, and Phantis, which are foodborne pathogens that can be transmitted in meats and vegetables, as seen here. To start, we should ask why antimicrobial resistance, or AMR, is such an important issue to study. This is because, according to the CDC, over 2.8 million people become infected and 35,000 people die from those infections in America alone every year. Furthermore, this number has been increasing since the last report from the CDC in 2013, which stated that at least 2.0 million people become infected per year and 23,000 people die. Therefore, this is a problem that has been increasing substantially in the past decade and will likely continue into the future. AMR is predicted to become much worse. This is a graph with current mortality for various diseases in blue and the projected AMR deaths in 2050 in purple. Currently, cancer kills over 8 million people, while it is estimated that 10 million deaths will result from AMR worldwide in 2050. This is why it's so important to determine ways to understand, prevent, and treat AMR. This is a diagram of how AMR occurs. Bacteria sensitive to a drug are in blue, and the few resistant bacteria who have genes that allow them not to be affected by the drug are in orange. When bacteria are exposed to the drug, sensitive bacteria die, leaving resistant bacteria to predominate. And on top of that, resistant bacteria can transfer their resistance genes to antibiotic susceptible bacteria and allow them to tolerate the drug as well. The more exposure that bacteria have to antimicrobial substances, the more resistance spreads. How are bacteria exposed to antimicrobial products? Some of the ways this occurred are pictured here. This includes medical exposure due to prescribing or overprescribing of drugs, or patients not taking the drugs as prescribed. Many antibiotics are also given to animals as a growth stimulant. Additionally, poor personal and hospital hygiene can lead to increases in AMR, and this is why it is very important to continue to develop new antibiotics, because a lack of new antibiotics is also a large contributor to antimicrobial resistance. I wanted to examine Salmonella strains since these are some of the most common foodborne pathogens. According to the CDC, Salmonella strains cause 1.35 million infections that lead to over 26,000 hospitalizations and 420 deaths per year in America. Salmonellosis is common and affects people of all ages, but the concerning thing is that infection rates are increasing. In the S. infantis that has been found to be multiply antibiotic resistant, this graph shows the relative rate in which a 1 indicates no change in rate and a less than 1 is a decrease, while a greater than 1 is an increase. So this shows an increasing trend since 2006. These infections are foodborne and usually begin with transmission from common foods such as poultry, eggs, and pork, or produce that are contaminated. Once the person has ingested a contaminated food item, symptoms will begin to appear around 6 to 72 hours after, and illness can usually last 4 to 7 days. Symptoms can include headache, fever, watery diarrhea, abdominal pain or cramps, and nausea or vomiting. But if the patient is dehydrated, this often causes further complications that can lead to hospitalization and death. Ideally, antibiotics will kill bacterial cells by disrupting key bacterial cell processes. This includes the synthesis of the cell wall, RNA synthesis, the production of proteins, or DNA replication. But bacteria that are not affected by antibiotics can use a number of strategies. They can use enzymes that inactivate the drug. They can exclude the drug either by decreasing membrane permeability or pumping out those antibiotics, or they can even change the target of the drug so that it no longer binds and has its effect. The genes that allow these bacteria to be drug resistant can also be transferred to other bacteria. And there are several ways that bacteria can transfer genes to one another, and the first is transformation, in which bacteria can take up free DNA. The second is transduction, in which genes are transferred through a bacterial virus called a phage. Lastly, in conjugation, genes are transferred through a sexpilus, through direct contact between the bacteria. Mobile genetic elements like plasmids, transposons, and integrons help to facilitate the transfer of genes. My project will focus on class 1 integrons, which are DNA elements that can spread antimicrobial resistance by essentially integrating and collecting multiple antimicrobial resistance genes in the form of these circular gene cassettes. Then transfer them between bacteria using the mechanisms of gene transfer like transformation, conjugation, and transduction. These integrons have 4 fundamental parts, the first of which is the Inti gene that encodes for the integrase, here in this purple. Integrase is a tyrosine recombinase that is needed for the recombination of the gene cassette into the integron itself. Next, there is the ATTI gene, which is the recombination site that is recognized by the integrase, so that is where the genes are integrated. Then there is the PC or promoter region that is the start of transcription of the gene cassette, allowing the genes to be expressed. Lastly, the pInt is another promoter region specifically for the transcription of the integrase. With all these parts combined, integrons can insert multiple gene cassettes with antimicrobial resistance genes, collecting them, then transferring them to other bacteria. Considering how class 1 integrons can mediate antimicrobial resistance in bacteria and that S. infantis strains tend to have multiple antibiotic resistance genes, I wanted to know if S. infantis AMR could be facilitated by type 1 integrons. So the goal of my research project was to examine 8 Salmonella enterica cerevar infantis strains obtained by the CDC that are resistant to multiple antibiotics and determine if any of these strains contain class 1 integrons. This is an overview of my project, and so to fulfill this goal, I would amplify an essential gene of the integron through PCR, then visualize any product of amplification using gel electrophoresis. Sequence analysis would then be performed, and BLAST, or basic local alignment search tool analysis, could determine if any amplified product is indeed S. infantis class 1 integron. My first step was to amplify the integron DNA using PCR, which stands for polymerase chain reaction. I designed primers to amplify the integrase gene Inti, which is this purple part right here on the integron mentioned before. Many cycles of denaturation or melting, annealing the primers to the DNA, and elongation result in many copies of the gene of interest. These can be visualized on an agarose gel. This is a diagram of gel electrophoresis, and the samples are loaded to the left, while electricity is applied to the gel. This allows the DNA to travel through the gel and resolve according to size, with smaller fragments traveling faster than large fragments. A ladder is then used to determine the size of the DNA fragments. This is an image of my samples after gel electrophoresis. The ladder is to the right and the samples are to the left. The bright bands are PCR products which were present in 6 out of the 8 strains. This PCR amplification and gel electrophoresis was repeated 3 times and suggested that 6 of the S. infantis strains have class 1 integrons. By comparing these bands to the DNA ladder here, they were shown to be 1.0 kilobase in size. Now I wanted to be sure that the PCR product was indeed part of type 1 integrons. To determine if the PCR products were part of type 1 integrons, I submitted these to the UT Sequencing Core Lab for analysis. I next analyzed sequence data using the basic local alignment search tool, or BLAST, and this confirmed that the gene I amplified was indeed the integron gene because they had an 85 to 98% similarity to documented sequences. My project thus determined that 6 of the 8 strains demonstrated class 1 integron with an 85 to 98% similarity to S. infantis. My next steps could include designing primers that can target the ATTI gene, this one right here, and extending the DNA to see what antimicrobial resistance genes are associated with the class 1 integron, since those gene cassettes are normally inserted next to that gene. And I could also compare the sequences for similar areas to see what areas might be conserved. I would like to thank the Centers for Disease Control and Prevention for providing the strains of Salmonella enterica cerevar-infantis, the St. Edward's University Department of Biological Sciences, Zoe Skelton, and of course, Dr. Bainham. Thank you everyone for coming to my presentation, and does anyone have any questions?
Hi everybody, thank you Angel, that was great. Trish, we'll start off with you and let you make some comments.
Okay, it was a pleasure to be able to mentor Angel. Since she first started at St. Edward's, she absolutely knew she wanted to be a physician, and she starts at UT El Paso in their medical school in fall. And I know she's going to be the kind of physician who reads all the current papers and keeps up with all the latest research and uses a really evidence-based approach to treat her patients. And so I know this is only the beginning of her wonderful career, and I hope that you'll stay in touch, Angel, and I'm giving you your stole and a virtual hug, and we can take a virtual image. Yeah, can you come up?
Yeah, okay, well,
Great job,
everybody
Angel.
sign on, because it's not great if they're all blank spots, you know. So sign on and I'll take a picture. We have plenty of time now, Angel. You stayed very well within the time period. Okay, ready?
Thank you everyone for coming.
Yeah, no, it's great. So any questions?
Oh, thank you for the well wishes, everyone.
I do have a question. Why did you want to research this? What was your motivation to research this?
I initially talked to Dr. Bainham about what research projects I could do, and she had mentioned that the CDC had given the university these 8 strains, and it would be really cool to see what we could do with them. So I looked into current research regarding Salmonella and antimicrobial resistance, because I had already done research with antimicrobial resistance with Dr. Bainham before, and so I thought it would be great to do something along the same vein. And so I found that there was a good amount of research done with specifically these integrons, and I didn't know what they were. So I looked into what they were, and they were really interesting. They're a large mediator for horizontal gene transfer, and therefore antimicrobial resistance spreading within Salmonella. So after I found out more about them, I realized it would be really fun to see if these strains specifically had that integron, and therefore would have that sort of mechanism for horizontal gene transfer.
No, I
got scared.
Go ahead. Were you done?
And it's been a great time. It's been an absolute pleasure to work with Dr.
Yeah, I'm a language professor, but I have to say with these presentations, I really love the language of science, that I get to hear something new and different and really fascinating. So you guys are great. If there are no more questions, we'll take another break. Andrew is coming up. Andrew.